Hello, science enthusiasts. Today, we have an exciting topic to dive into: AI-driven citizen science and recombinant adeno-associated virus drug formulation development with this assistance of ChatGPT. Sorry for the long title. If you are watching this, I am going to assume that you have a high-level understanding of ChatGPT. Before we get started, let's first understand at a basic level what recombinant adeno-associated virus drug products are all about. Recombinant adeno-associated virus, or RAAV, is a type of virus used in gene therapy, a cutting-edge field that holds great promise for treating genetic diseases. RAAV drug products are customized formulations that carry therapeutic genes to target cells in the body, aiming to correct or replace faulty genes responsible for diseases. Developing these drug formulations is a complex and time-consuming task, requiring years of expertise. Traditional methods involve running numerous experiments to test different formulation conditions, making it an arduous process. With the help of ChatGPT, an AI language model, I embarked on a proof-of-concept project to demonstrate the power of AI-driven citizen science in RAAV drug formulation development. Quickly, what is ChatGPT? It's an AI language model created by OpenAI that can understand and generate human-like text. It is particularly good at assisting with software development coding tasks. With its assistance, I was able to rapidly prototype an artificial neural network, or ANN, to predict the lethality of different RAAV drug formulations. The collaboration with ChatGPT was truly game-changing, especially for someone like me, a wet lab biologist with limited AI and coding experience. Hypothetically, I would be able to focus on the formulation development while ChatGPT handled the complexities of AI-driven methodologies. Now, let's talk about citizen science. Citizen science is all about involving non-experts, like you and me, in scientific research. There are a variety of citizen science projects open for the public to participate in a variety of ways. Citizen science projects include Zooniverse, Starter, and Project Budburst. With AI-driven solutions, citizen scientists can actively participate in drug development and biopharmaceutical research with limited domain knowledge. I must emphasize that this project is a proof of concept. The model's current accuracy stands at around 54%. Validation with real-world experimental data is crucial for further improvements. Let's go over this project's methodology. First, I generated a synthetic dataset in Python using the pandas, NumPy, and random libraries. I did this because there was a lack of publicly available RAAV drug product formulation data. I could explain the synthetic data generation process in another video if people are interested. You can check out the code for that by following the git.hub link located in the description. This dataset had numerical data types for vector concentration, cryoprotectant concentration, lipoprotectant concentration, buffer pH, buffer concentration, and preservative concentration. There were also categorical data types for cryoprotectant type, lipoprotectant type, buffer type, bulking agent type, preservative type, and lethality. Then I did research on what machine learning models have been used in gene therapy drug product formulation development. I found a variety of papers that referenced using AI models to assist in the drug development process. With some prior knowledge of AI, I thought to use an artificial neural network, as it is able to take in a variety of variable types and is often used to make predictions based on training data. A quick explanation, high-level explanation of an artificial neural network is a type of machine learning model inspired by the human brain, mimicking the way that biological neurons signal to one another. As an alternative to following another online tutorial, I turned to ChatGPT in order to generate the skeleton of a Python script that I could work off of. I knew that the neural network needed to have certain qualities that coincided with the synthetic dataset I had generated. Therefore, I tried to mention those qualities in the first prompt that I gave to ChatGPT. The first prompt I fed to ChatGPT was, "Help me come up with an artificial neural network in Python that helps me predict the outcome of different formulation of an RAAV drug products." The neural network will predict if the combination it reads will be lethal or non-lethal. The data used to train this neural net will be a data frame with the variables of vector concentration, cryoprotectant type, cryoprotectant concentration, lipoprotectant type, lipoprotectant concentration, buffer type, buffer pH, buffer concentration, bulking agent type, bulking agent concentration, preservative type, preservative concentration, and lethality. The neural network is trying to predict lethality. The numerical variables are vector concentration, cryoprotectant concentration, lipoprotectant concentration, buffer pH, buffer concentration, and preservative concentration. These start off as float data types. The categorical variables are cryoprotectant type, lipoprotectant type, buffer type, bulking agent type, preservative type, and lethality. The first thing I need to do is pre-process the data so that it can be more easily understood by the artificial neural network. Next, I need to build and train the neural network. Can you provide me with some code to accomplish this? I was sure to be nice to save myself during the AI revolution. ChatGPT was able to quickly generate a response in several code blocks with explanations for what was going on. Initially, I asked ChatGPT to explain the different layers included in the neural network and how to optimize them myself. The answer implied that trial and error was needed, and I did not have time to go through all of that. Therefore, I asked ChatGPT to help me change the code so that I could automatically try different layer feature combinations. ChatGPT called this process hyperparameter optimization. The code required changes from there that I was able to guide myself through without turning to Stack Overflow or a YouTube tutorial even once. I fed my code with the changes I made to ChatGPT and asked it to add comments to make it easier to understand. Unfortunately, even with the hyperparameter optimization, I was only able to get an accuracy score of just above 50%. I chalk this up to the synthetic data needing to be peer-reviewed before being used in this manner. This process overall took under 2 hours of commitment, a feat I would consider impressive considering that I have limited coding experience. That is, if the code actually works on real-world data. I wasn't done with ChatGPT, as I then asked it to help me come up with the Python Markdown file displayed on the project GitHub page linked below. I also asked it to help come up with this very script in the LinkedIn post I used to announce this project. With ChatGPT, I truly felt like I have my own research assistant. Enough free promotion. I hope this was a helpful video for all those interested in citizen science coding projects or those in the gene therapy space. I hate to say this, but comment, like, and subscribe if you are interested. I would love to make more content about citizen science projects. Have a great day.